Hello, bachcho. Let's see the question. So the question says the thermostable enzymes Taq and PFU isolated from thermophilic bacteria are. So इन चारों options में से हमें Taq and PFU के type को identify करना है. That is what type of enzymes these Taq and PFU actually are. So first of all, the key concept in this question is to understand what are Taq and PFU. So first of all, Taq and PFU are enzymes. That is, they catalyzes some particular metabolic reaction. So this enzyme is actually a type of DNA polymerase. So Taq and PFU are types of DNA polymerase enzyme. Now the enzyme Taq polymerase is isolated from the bacteria Thermophilus aquaticus, and the enzyme PFU polymerase is isolated from Pyrococcus furiosus. Now, both these bacteria are thermophilic bacteria, which means that the optimum condition of living for these bacteria are very high temperatures. So these bacteria are archaebacteria and they are mostly found in extreme conditions of very high temperature. So these enzymes, that is Taq polymerase and PFU, are isolated from these thermophilic bacteria. Now, since Taq and PFU polymerase are isolated from the thermophilic bacteria, therefore these enzymes are also thermostable enzymes. So thermostable enzymes are those enzymes which can withstand very high temperatures of around 94 degrees centigrade as well. So most of the enzymes get denatured at very high temperature, but the 3D structure of the Taq and the PFU polymerase enzymes are not destroyed at a very high temperature, but they can withstand high temperatures because they are found in thermophilic bacteria. Now, Taq and PFU polymerase play a major role during DNA replication because they can synthesize, or they are responsible for the synthesis of new DNA strands, and therefore both of them are types of DNA polymerases. So Taq and PFU are types of DNA polymerases. Now both these enzymes play a major role during the polymerase chain reaction, or the PCR. So Taq and PFU are used as DNA polymerases for the process of PCR, because in PCR we require high temperatures, and these enzymes, since can withstand high temperature, therefore they are used for the DNA amplification by the synthesis of new strands of DNA complementary to the template strand is done by Taq and PFU during the polymerase chain reaction. So now if we see the options, then the first option says RNA polymerases. So they are not types of RNA polymerases. Second option says DNA polymerases. So DNA polymerase is the correct answer because Taq and PFU synthesizes new strands of DNA and therefore they are DNA polymerases. The third one is restriction endonucleases. So again, this is incorrect because they do not cut the DNA fragments, but they synthesize new strands of DNA. And the fourth one is DNA ligase. Again, it won't be ligase because they do not join two fragments of DNA, but they synthesizes new fragments of DNA. So therefore, our final answer for this question is option number 2, that is DNA polymerases. So Taq and PFU are type of DNA polymerases. Hope you understood this well. Best of luck. Thank you.